If you are using a screen reader, please be aware that the controls are at the bottom of the Zoom interface. This control bar may collapse when it is not in use. If you prefer to prevent the controls from auto-hiding, go to Settings within the Zoom platform, select Accessibility, and then select Always Show Meeting Controls. It might be helpful to maximize your window and navigate by using the Tab key. Additional buttons and settings are available by pressing Alt. During the call, you may ask questions through the Q&A and the chat features. We will address the questions towards the end of the call during the Q&A session, at which time we will provide additional instructions on how to ask your questions. I'd like to turn it back over to Steve now.
Thanks, Kelly. I appreciate it. I just wanted to welcome everybody to the program today. There's still people joining, and we have a really good group out here. I wanted to thank the Medical College of Wisconsin Eye Institute for being a partner of the foundation for many years, since the first walk that we did almost 15 years ago. And it started out as a small little chapter meeting in a library in Milwaukee, and it has grown to one of our bigger walks in the country and one of our bigger fundraising areas in the country. The foundation has been around since 1971. Our mission is our urgent mission, and we hold true to that. We are focused on finding preventions, treatments, and cures for a whole host of retinal degenerative diseases, which include retinitis pigmentosa, also known as RP, macular degeneration, which is a eye disease which is familiar to many of you. It affects a big part of our population, and specifically our older generation usually. Usher syndrome, which is a combination of hearing and vision loss, choroideremia, retinoschisis, Leber's congenital amaurosis, which is a big one for anybody on our call tonight that does not know much about research. That would be a great one to look up. It's called LCA, Leber's congenital amaurosis. It is a disease which actually the foundation has funded research that has brought treatments and cures for that specific gene, RPE65. And that's a really good story to find about what the momentum is in the world of research. I've been with the foundation for 14 years now. I absolutely love what I do. I love working for the foundation and working with our families across the country. I spent a lot of time in Wisconsin, both as part of my job and also fishing and dropping a lot of my money off at Potawatomi Casino. So you guys should be thankful for me for a lot of different reasons. But I love Wisconsin and working with two great institutions. We're going to focus today with our partners from the Medical College of Wisconsin. The Eye Institute has been a great partner with ours, both in hosting meetings in person, but also McPherson Eye in Madison. Dr. David Gamm as well. I'd be remiss to not mention that group as well. The groups both work together very well with sharing information, and the foundation has doled out grant money in some of our years in excess of $1 million split between both of the institutions. So the money, and I can say this thankfully because sometimes I can't say that in other states that maybe don't have the best research. The money that is raised in Wisconsin stays in Wisconsin, and then tenfold, because our walk raises about $100,000, and I'd say every year we're close to $750 to $1 million that stays right in Wisconsin. So there's a lot of great research that you guys should be very proud of, and you'll hear some of that today. With that, I'd like to pass the microphone off to Dr. Deborah Kostakis, who is the fairly new chair and professor, Department of Ophthalmology and Visual Sciences at the Medical College. And Dr. Kostakis was also our medical chair at our walk, as we talked earlier. She was there in the deluge of rain a couple years ago, and we're hoping that she returns again in some nicer weather this year. So Dr. Kostakis, take it over.
Thank you. It was arguably hail and maybe even snow, but today is a much nicer day. It's too bad we're not walking, but good evening to you all. And thank you for that introduction, Steve. I understand that in addition to FFB, there may be people here from the Lions Club. And so welcome to all of you. I think we have some really exciting projects to share with you. FFB, as Steve mentioned, has been really instrumental in helping the Eye Institute researchers discover And we all know that that's what's needed in order to go to the next step to develop treatments and cures. And they've been partners all along the way. So we very much value your partnership. Also, I need to thank Julianne and Kelly and Gina, who really managed to get this great program together. So without further ado, let's move to hearing from our VIPs of the evening, our researchers.
I'd like to welcome Daniel Lipinski, who is assistant professor at the Medical College of Wisconsin. Dr. Lipinski?
Good afternoon. My name's Dan Lipinski. I'm assistant professor of ophthalmology and director of the Ocular Gene Therapy Laboratory at the Medical College of Wisconsin. And I'm going to be talking to you a little bit about some of our work on the development of gene therapies for rare inherited retinal disease. As an FFB chapter meeting, I'm very aware that many of you in the audience will either have an inherited retinal disease or know friends or family that do. And so you will all be very, very knowledgeable about the eye and the different cell types in the eye. Before we're going on, I did just want to mention that inherited retinal disease are typically caused by genes that are expressed in either the photoreceptors, the light-sensitive cells at the back of the eye, or the retinal pigment epithelium, which is a layer of cells that sits under the photoreceptors and helps keep those cells healthy. And that in inherited retinal diseases, vision loss is primarily the result of either the degeneration or dysfunction of the photoreceptors themselves. There's a huge unmet clinical need to develop therapies for inherited retinal disease. There's really an absence of both gene therapies, cell therapies, and pharmacological treatments for these diseases. And as such, it's an area of really active research, generously supported by foundations such as FFB, the federal government through the NIH, as well as philanthropically from patients themselves. The disease I'm going to focus on today is retinitis pigmentosa, which collectively affects about 1 in 4,000 people worldwide. Now, RP is a genetically and phenotypically heterogeneous disease, which essentially just means it can be caused by mutations in many, many different genes, and it can present in the clinic in a number of different ways. That being said, the most common presentation of RP is as a rod cone dystrophy. And this is where rod photoreceptors, which mediate low light vision and contrast sensitivity, degenerate first due to an underlying genetic abnormality. And this is then followed by the secondary loss of cone photoreceptors, which mediate daytime vision and color perception. And RP is a challenging disease to treat for a number of reasons. Firstly, the mechanism by which cone photoreceptors degenerate is incompletely understood. These cells are genetically healthy and they should survive, but inevitably, once a certain proportion of the rods have been lost, the cones will follow along through a process we don't understand. Secondly, treatment is also challenging due to the oftentimes late presentation of the disease. In our kind of electrically lit world, people don't really notice that their nighttime vision is getting worse, and so only really tend to turn up to the clinic once they're starting having difficulties reading or to driving or to recognize objects or color looks different. At which point the patient has already lost a lot of rod photoreceptors and is already losing their cone photoreceptors. So there's quite a narrow window in which we can intervene to keep those cells alive and to maintain vision. The last reason is that in approximately 30% of recessive cases and 50% of dominantly inherited cases of RP, we don't know what the genetic cause is, which makes a traditional approach to gene therapy impossible, whereby you would simply replace the gene that you know to be missing. So this brings us to the question of how exactly do you treat something that you don't understand? And so in an early stage of disease, when a patient still has vision, the priority has to be simply to preserve whatever cells remain in that patient's retina. And one strategy that our laboratory and many others around the world are taking to do this is through something called neuroprotection, where we essentially just modify some of the remaining cells in the retina to produce a supportive molecule that acts to prevent or delay cell death, particularly of the cone photoreceptors, which are so critical for daytime vision. The benefits of this approach is that it's so-called gene independent. We don't have to understand what the genetic abnormality in that patient is in order to preserve cells. So the real question is, does this approach work? And so I'm just going to share with you a little bit of the data we've been generating over the years on something called ciliary neurotrophic factor, or CNTF. And so to test out, does a therapeutic work, we first need a model. So one of the great resources that we have in the ocular gene therapy laboratory is a number of different animal models of retinal disease. And one of the really unique ones that we have access to is a mouse model where all of the cone photoreceptors are green fluorescent. And this is really, really critical because it allows us to very easily see these cells and to see how many of those cells are surviving in response to a therapy. And so we have two versions of this model. We have one that has a normal retina. So these green fluorescent cone photoreceptors survive throughout the lifetime of the animal. We also have a retinitis pigmentosa version of this model, where just like in the human patients, there's an abnormality in the rod photoreceptors. Those cells degenerate by about 8 to 12 weeks, and that's then followed by the secondary loss of these green fluorescent cone photoreceptors. One of the amazing benefits of this animal model is that by having fluorescent cones, we can very easily visualize these cells in real time, in real life, using many of the same imaging techniques that some of you may have experienced during your trips to the optometrist and the ophthalmologist. We can non-invasively image the retina very easily. And in these animals, what you see is this beautiful kind of starry night scene when you look in the back of their eye. And each one of these little white dots on this top left-hand panel is an individual cone photoreceptor. And that's amazing because it means that we can literally look in these animals' eyes and count how many cones are surviving in response to our gene therapy treatment. And so going back to the question of, does neuroprotective protection work? Yes, it can work. So what we did in one study is we took a modified virus vector to deliver the gene for CNTF to cells of the retina in this animal model. That CNTF is then produced and it's secreted throughout the retina. And what that leads to is a very, very robust preservation of cone photoreceptors following a single dose of our virus vector. What this looks like is, on the bottom here are two eyes from the same animal model. On the top, you'll see there's lots of these green fluorescent dots, each one of which is a surviving cone photoreceptor. And as this animal gets older and older, all the way out to about 7 months, you'll see lots of these dots are still remaining. And the retina, in fact, looks pretty normal. In the untreated eye in this animal, you'll see again, there's lots of green dots to start with, but those fluorescent cone photoreceptors progressively degenerate. And by about week 18, they're almost all gone. They've lost their cone photoreceptors. And by 7 months, their retina is now looking extremely, extremely abnormal. So this is really strong evidence that neuroprotection can be very, very effective at preserving cone photoreceptors if you can treat these patients or these animals early. When we take a bit of a deeper dive and look at the histology in these animals, we see an identical picture. So in our treated eyes, at the top here, we're seeing lots of green fluorescent cone photoreceptors surviving all the way out to 7 months post-treatment. Whereas compared to the bottom panel, where these eyes have not been treated with our CNTF gene therapy, you see that all of these fluorescent cells have now degenerated, and those eyes have lost all vision. Which really brings us to the question of, if CNTF is able to preserve cells so well, why isn't this a therapy that is currently available? Why can't you go to your ophthalmologist and ask to have CNTF gene therapy? And the answer is, unfortunately, that whilst CNTF is extremely, extremely effective at keeping those cells alive, it has a negative effect whereby it suppresses their function. So the cells are present, but they don't work, and they would be unable to relay, useful visual information back to the brain. And so what we wanted to do, building on this study that shows neuroprotection can work, we can preserve those cells, can we separate out the protective effects of CNTF from these functionally suppressive effects? And we were absolutely delighted to receive an individual investigator research award from Foundation for Fighting Blindness to support some of this work, where we try and tease apart the different effects of this neuroprotective molecule. And this is ongoing research, and I would be absolutely delighted to answer any questions you have about that research at our Q&A session later, or alternatively, I'm very, very happy if you would just like to send me an email or give me a phone call, and I'm always delighted to talk to patients and their families. And with that, I'd just like to finish by acknowledging all of the many amazing staff and students in my lab, as well as all the funding agencies from the NEI, FFB, Glaucoma Research Foundation, McPherson Eye Research Institute that support all of our work and all of our people, all of the many collaborators we have worldwide helping us out developing these therapies. And I would just like to thank you for your time and your patience. And again, I'll be very happy to answer any questions you have later on.
Oh, hang on. Can you guys hear me?
Yes.
Yeah.
All right, very good. Thanks so much, Dr. Lipinski. I just want to let you guys know, for anybody else that has joined us as well, the brief presentations are actually recorded just for the smoothness of the transition. All the doctors are on with us live today. I just wanted to say that because I know this is kind of a new experience for all of us with Zoom. So there's a couple glitches that were caused during his presentation that I actually caused myself. So I apologize for that. Again, thank you, Dr. Lipinski. I'd like to now move on to Dr. Iris Kassem and her presentation on improving the outcomes for children after cataract surgery. Dr. Kassem?
A little background on myself. I'm a fellowship-trained pediatric ophthalmologist with several special interests that guide my research, and vice versa. The clinical focus related to today's talk is pediatric cataracts, such as this child who had a cataract in his left eye. He's much older now. Even though everything was perfect with the surgery, he has experienced several complications over the years, secondary to the surgery, that resulted in some difficulties with his eye. Hopefully with science, though, we can discover new therapies so that doesn't happen in the future. Before I get started, I just want to acknowledge the village that has helped move this project forward, including lab members, collaborators, and funding sources. In particular, I want to acknowledge Jonathan Young, who is a graduate student finishing up his PhD thesis in the lab, and most of this work is his work. So pediatric cataracts is one of the most common reasons for pediatric intraocular surgery. Usually the pupil is black, and that's because the lens is clear, but sometimes children and adults get a cloudy lens that can make the pupil cloudy or white, like in this child with bilateral cataracts. And the picture below that is a closer-up picture of an eye of a child who had cataracts from the time they were born. In children, it's unique because they could develop something called amblyopia if you leave it untreated, so time is of the essence. Amblyopia is basically a decrease in the best corrected vision of an eye that would otherwise look normal, or if there's some abnormalities of the eye, the vision that they have is worse than you would expect given their problems. And that's because there's a critical period for the vision to develop in children up until about the age of 8 years old. If they don't have normal vision during this time and you don't correct things like cataracts or blurry vision, that vision can be reduced even after that problem is corrected and it's permanent. So in children, when they have cataracts, removing the cataract makes the vision outcome better. The problem is if you have like a newborn with a cataract, they have a very exaggerated response to surgery compared to adults. So in an adult, you'll remove a cataract and you'll put in an artificial lens to focus the vision. But in children, a lot of people avoid placing an intraocular lens at the time of surgery because that could result in a lot of complications. So surgeons have two options. One is to remove the lens and leave it what we call aphakic, so we don't put in an intraocular lens. It's difficult because fitting a contact lens is hard in children. The eye's always changing shape and growing, and it could get infected. It could be impractical because somebody else has to put the lens in and out and clean it, or maybe where they live just isn't ideal because of the risk of infection. And if you don't use the contact lens, if they're aphakic, especially with one eye being aphakic, not using the contact lens can result in amblyopia because the vision's blurry because they don't have the intraocular lens. But on the other hand, if you implant the intraocular lens, they could get severe inflammation, they could get scarring, they could get a white membrane, such as in this picture, and the pupil can be displaced also as in this picture, and it could be severe enough that it blocks the vision altogether, also causing amblyopia. So our lab works on the rationale that it could potentially be advantageous to use intraocular lenses in the eye of an infant and get better vision if we could decrease this inflammation and scarring, because it would constantly provide that focus for the eye. It would decrease the risk of infection. It would decrease the burden on the family significantly, and the goal of our lab is to characterize the response after surgery using both humans and animals. We don't just rely on human subjects because they're very variable. Each person is completely different. Pediatric surgery is relatively rare, and there are different pre- and post-operative regimens that you may use for a particular patient, so that makes things very variable. So animal models are really useful. They're often used in industry. In the case of intraocular lenses, New Zealand white rabbits are very common to use. The eye size for the portion of cataract surgery is very similar to humans to the point where we use the same instruments, the same surgery, and they also have the same exaggerated immune response, even more than in humans. So the picture on the left is a human after surgery, the right is a rabbit, so they have a lot more of that fibrin, and they have inflammation. So our thought is if we could get a rabbit to have a good postoperative outcome, humans will probably also do very well. Our hypothesis goes in that we think that some proteins increase after surgery or appear, and some proteins decrease after surgery, and those proteins influence the postoperative response. What we did initially was we broke down the surgery into 4 different steps, and we looked at the changes in the proteins at each one of these steps using mass spectrometry. And the graph on the bottom right is an example of some of our results from mass spectrometry, and we break these down to filter out what proteins are in the fluid of the eye and what abundance there is. So we look at the fluid before surgery and after surgery, compare those, and see the protein changes in response to the procedure we do, one of these 4 steps of cataract surgery. So just in brief, we had some interesting results. The further along we went in surgery, especially when we put that intraocular lens in, the more inflammation and scarring we saw. And in terms of proteins, we saw that there were 2 major categories of proteins that increased, one related to blood clots or fibrin formation, coagulation cascade, and the other one being related to inflammation, the complement inflammatory cascade. Interestingly, the immunosuppressive proteins, some of them decrease significantly after procedures, so that's a future arm to target for therapy. But looking at the coagulation cascade, how much it increased, we thought we could use a blood thinner perhaps to decrease complications after surgery and scarring. So what we did was we would do the surgery, and then we would inject a candidate medication like Lovenox or enoxaparin, which is a blood thinner. with a long half-life, and we would see what it looked like after surgery. So this is what it looks like untreated. So we just do cataract surgery, implant the IOL, and we would get a huge immune response and lots of scarring. Then we would put, at the end of surgery, we would inject a large dose of Lovenox or enoxaparin. And you could see compared to the untreated, there really isn't much of that scar, or that fibrin clot is what we call it in the lab. And that's probably because it's interfering with the coagulation or blood clotting cascade, which is a great result. We also looked at triamcinolone, which is a steroid, we use in the eye already to prevent inflammation. It didn't have a huge response or a huge improvement, but it did have some improvement post-operative response. But then when we combined those two drugs, we really had a very nice looking eye after surgery. You could even see the intraocular lens clearly. The eye has something called a red reflex, which is really nice. So we're pretty hopeful that maybe this may be something to use in the future. They have done some studies on humans looking at enoxaparin in the eye, but the dose they used was pretty low. So there's more to be done before we bring it to patients, but it's pretty hopeful. We also looked at treatment therapies, given that there was the whole clotting cascade. We looked at a clot buster, tissue plasminogen activator, and that basically breaks the clots in the brain if you have a stroke or a heart attack. And we thought maybe it could break up our fibrin clots in the eye after surgery. So this is a couple of eyes with just surgery without treatment, 3 days after surgery. So you see those large fibrin clots in the front of the eye. We injected a control or just saline solution, and 24 hours after we injected that, not much of a change, maybe a little better, but nothing significant. And then when we injected that clot buster, TPA, there's much less of that fibrin scar in the front of the eye. So we're hopeful that that may be a treatment in the future to be able to use in patients that have a lot of scarring after surgery or with some other complication. So in conclusion, enoxaparin is promising to prevent fibrin formation. And when used in combination with the steroid, it could also decrease inflammation. So working on two parts of the postoperative response. And TPA improved the clarity of the eye after the scarring had already occurred. Things we're looking at in the future are looking at new treatments based on those protein abundance changes we saw with mass spectrometry. So we're looking at inhibiting some proteins that are more abundant after surgery. We're looking to replace some of those that decreased after surgery. And we're also looking at non-steroid treatments that may be used as eye drops. In addition, we're looking at the effects of those treatments that we studied, and we're looking at correlating it with human samples. And we actually have a large study trying to collect as many human samples as we can. So that's some of our research we do in the lab, and these are some of the references for our research. And if you have any questions, please feel free to reach out. Thank you very much.
Thank you, Dr. Kassam. I appreciate that. I would like to now move on to Dr. Collery, who is the assistant professor at the Medical College of Wisconsin. He will be talking about the leveraging of zebrafish to study eye diseases associated with the visual cycle. Dr. Collery.
Good evening. My name is Ross Collery, and I am a member of the research faculty at the Medical College of Wisconsin Eye Institute. I'd like to thank you very much for this opportunity to speak to this meeting of Foundation Fighting Blindness. I admire the mission very much, and I have benefited greatly from working with people who have been funded by the generosity of your donors. Today, I'd like to give an overview of how I use zebrafish to try to better understand blinding disorders that affect human patients, especially those caused by mutations in the visual cycle, and to explain how I can leverage these models to help develop and assess therapies. So in a nutshell, my work focuses on components of the visual cycle and their role in retinal degeneration and refractive error. So what do I mean by that? Well, photoreceptors at the back of the eye have an extremely high requirement for vitamin A and related molecules, and have a special pathway devoted to vitamin A transport from the circulating blood supply. Mutations in any of the genes that make up this pathway lead to retinal disease and vision loss, but not always the same type of vision loss or with the same degree of severity. This pathway is called the visual cycle and is necessary for retinal health and eye size control. Understanding how the components of this pathway function in the eye is critical to better treatment of eye disease and vision restoration. You should be able to see that each component of this pathway has a different blinding disease pathology associated with it. And part of the research that I undertake is to understand why this is the case, how these diseases progress, and what we can do to help them. So I use zebrafish as a model organism to study eye diseases and refractive error. The reason that I do this is that zebrafish are small and cost-effective. They have colored daylight vision, as we do, and we have powerful gene-modifying tools available in my lab. We're able to precisely insert or remove genes as desired. The chasm between human and zebrafish genes is not as great as you might think, and almost all genes associated with a human disease have a zebrafish counterpart. The cone photoreceptors in the zebrafish retina are laid out in a neatly ordered array, and their abundance is similar to that of the human parafovea. We're able to easily visualize the cone photoreceptors in living fish using apparatus from Dr. Joseph Carroll's optical coherence tomography program. And in the set of panels on the right, each point of light in panels B and C represent an individual cone photoreceptor, and their number and health can be measured and quantified using optical coherence tomography. So we use approaches that are either transgenic or mutant to insert or remove genes from zebrafish. We can use transgenic techniques to insert novel genes and express them in any cell of our choosing, for example, cones or rods. On the series of panels on the left, you'll be able to see that we can inject zebrafish eggs and insert a fluorescent gene, and we can replace that fluorescent gene with any gene that we would like for therapeutic reasons. Conversely, we use CRISPRs, the molecular scissors, to cut out pieces of genes or change them by a single letter to mimic human disease mutations. In the panel on the bottom right, you'll see an example of where I've used 2 CRISPRs to completely remove a disease-causing gene and some DNA sequencing data that verified that I had indeed inactivated that gene by its deletion. So how can we use zebrafish to learn more about vision? In the upper left panel, you can see that I've been able to generate both large-eyed and small-eyed fish. Myopic and hyperopic human patients can suffer from disease pathologies of the retina, and I've been able to find out a little bit more about how that happens. I mentioned the use of transgenic tools. You can see that zebrafish here are expressing fluorescent proteins in their cones and their rods. We've been able to use CRISPRs to inactivate genes of our choosing. In this example here, CRISPRs are being injected into a zebrafish egg that's resulting in the loss of a pigmentation gene. I've developed a system whereby I use optical coherence tomography to measure aspects of the eye, and by measuring the size of the lens and the axial length of the eye, I can calculate the refractive error of that eye, and I can reasonably say whether or not the zebrafish eye is correctly focused or not, as the case may be in a disease phenotype. Similarly, I can use optical coherence tomography to look closely at the retinal lamination and to count and quantify the individual cone photoreceptors in the fish eye. And then finally, I'm able to carry out visual testing where we can present a visual stimulus to a fish and see whether or not that fish is able to respond to it. This fish on the bottom is normally sighted, and its eyes move to track the striped stimulus, where the fish on the top is completely blind and is unable to respond to the visual stimulus. My lab has already generated multiple disease models in visual cycle genes. So, for example, mutations in STRO6 cause retinal disease in humans, and our zebrafish model shows loss of rods and unhealthy cones. On the left, you can see that the STRO6 fish has lost many of those points of light that correspond to photoreceptors. And then using more traditional histology, we can see that the photoreceptors in the STRO6 column are much shorter and or completely missing. Another mutant that I've developed, mutations in human IRBP cause retinitis pigmentosa and myopia, which we've successfully modeled in fish. I've been able to show that IRBP mutant fish develop myopia at 4 weeks of age. So now I'd like to discuss another blinding disease that's caused by a mutation in a gene called ABCA4, and that is Stargardt disease. This type of macular degeneration affects young people and has no therapy available. It onsets in young people and it gets worse as time goes on. It's caused, as I said, by mutations in a gene called ABCA4. It's considered a type of macular degeneration, and it's caused by inappropriate accumulation of a molecule called retinol in the eye. So I set about making a Stargardt disease zebrafish model. Normally, what ABCA4 does in the eye is export all trans retinal from the photoreceptor before it can merge with other molecules to become a toxic molecule called lipofuscin. This causes damage to the retina, resulting in vision loss, and that is the basis of Stargardt disease. Currently, there are mouse and dog models, but those both come with their own difficulties to use. Zebrafish actually have two ABCA4 genes. One is expressed in the RPE and retina, and one is in the retina alone. And I've used CRISPRs to inactivate both genes. I'm in the process of merging both mutant lines in order to completely inactivate both ABCA4A and ABCA4B. And I'm very excited to see how closely the human disease is paralleled in this cone-rich zebrafish model. As well as trying to generate a disease model, I'm very interested in trying to develop potential therapies that might provide some usefulness and insight into restorative approaches for people with Stargardt disease. So my first therapeutic approach that I'm working on is to express a naturally occurring protein, RDH8, that is part of the natural visual cycle and converts all trans-retinal to all trans-retinol, converting it from a toxic to a benign form. So I've already started expressing that in the cone photoreceptors of zebrafish, where I've seen it causes no damaging effects, and I'm excited to see whether or not that will be useful to treat the Stargardt disease fish I'm making. One of the difficulties with using gene therapy to treat Stargardt disease is that the gene itself, ABCA4, is extremely large. My second therapeutic approach is to make a synthetic retinol transporter. I've taken the critical retinoid binding region of ABCA4 and put it into another gene that's naturally found in the eye. I've hollowed out that gene essentially, and I've merged it with the critical part of ABCA4. And I've found, similarly to my first approach, that I can express it without any harm in the zebrafish retina. And so I'd like to see whether or not that has any restorative properties for the Stargardt fish. These are my genetic approaches, but I also think it's necessary and important to pursue pharmacological therapies that might be useful for Stargardt disease. So this is a schematic of the visual cycle, and you can see that there are a couple of intervention points that people have already used to try and prevent the inappropriate accumulation of retinoids. One here is a mixostat, which blocks RPE65 and prevents more retinoid being fed into the cycle. Another similar approach is using fenretinide to prevent the ingress of retinol from the bloodstream. What I'm doing is developing an in vivo retinol biosensor so that I can test out pharmacological approaches to see whether or not retinal can be converted to retinol by any of these techniques. And so what I've done is I've cloned the RBP4 gene, which naturally binds all transretinol, and only when this binding takes place can it recruit a third protein, second protein, I guess, that's called transthyretin. These two proteins actually form a tetrameric complex. This large four-protein complex has evolved in order to prevent its loss through the kidneys as it circulates. To the RBP4 molecule, I've cloned what I think of as a part of a light bulb. This is the filament, and then the other part of the light bulb I've attached to transthyretin. And only when these two proteins come together, which, as I say, only happens when all transretinol is present, will that light bulb give off a fluorescent signal. This approach, this bimolecular fluorescence complementation, works in both tissue culture and in zebrafish. I'm happy to be able to show that some of the cell culture experiments that I've been using show that it's a very good biosensor for the accumulation of retinol in cells, and I'm sure that it will be also in tissues. So it only remains for me to give you my acknowledgements. I want to thank everybody who works so hard in my lab. I really appreciate their hard work and dedication. I'd like to thank all of the other labs at the Eye Institute and elsewhere at MCW. All of the vision research labs are wonderful people to bounce ideas off and to interact with. I'd especially like to thank Joe Carroll and the current and former people in his lab who've been so very helpful with optical coherence tomography techniques. I'd like to thank everybody at the Eye Institute, including the people who make it so very possible to carry out these great communication programs, and also the people who make all of our research facility work so smoothly. I'd like to thank my current research from the E. Matilda Ziegler Foundation for the Blind, and I'd like to most of all thank you all and invite you to ask me anything at all. Thank you.
Thank you, Dr. Collery. I'll tell you what, I wanted to just make a comment because all the speakers have ended their presentations with welcoming you to ask questions and be open about that. I work with a lot of doctors and institutions across the country, and I'll tell you, the Medical College of Wisconsin Eye Institute, the doctors there are so open to, they want you guys to see what they're doing, what they're working on. And that's the same approach with all our doctors across the country, with focusing on bringing treatments and cures for our families. That's the biggest thing. But I'll tell you what, it's nice to have doctors that are very friendly and want to share their experiences. Joe Carroll in that lab and back with Dr. Connor as well. Couldn't be nicer, couldn't be better to work with. And that's not a sales pitch. That's not I know it sounds corny, but it's really a pleasure to work with that kind of group, and to know that they are solely focused on bringing treatments and cures for your retinal degenerative diseases. So I just wanted to throw that out there before we introduce our next speaker. But so again, thank you to Dr. Collery. Now I'd like to move on to our final speaker, and then we'll move on to our Q&A. Please welcome Dr. Ahmed, who's another assistant professor at MCW, who'll be talking about artificial intelligence in ophthalmology. Dr. Ahmed. Wait, what did I do? Ignore that.
Good evening. I'm Basir Ahmed from the Department of Ophthalmology at the Medical College of Wisconsin. I'd like to speak to you tonight about how artificial intelligence has made inroads into ophthalmology.
Hello, Hal, do you read me? Do you read me, Hal?
Affirmative, Dave. I read you.
Open the pod bay doors, Hal.
I'm sorry, Dave. I'm afraid I can't do that.
What's the problem?
I think you know what the problem is just as well as I do.
Well, that was a clip from the famous classic movie 2001: A Space Odyssey. And AI is something that people have dreamed about for decades. It really generally and is defined as mimicry of human performance at tasks and getting better over time. There are two types of AI that have been postulated: narrow AI and general AI. General AI is kind of science fiction. It's the flexible approach where machines become self-aware and get better at doing different tasks and learn new tasks and things. But in reality, what does exist today, and actually exists all around us, whether we realize it or not, is narrow artificial intelligence. And examples of that include speech and language recognition, things like Alexa that you might have at your house, image recognition when you put up something on social media and people get tagged automatically in those photos, your phone when it's suggesting points of interest or alternate routes going home using Google Maps, or your phone putting certain applications to sleep based on your usage or behavioral patterns. Those are all examples of just how prevalent AI has become. So some of those areas in which AI has shown practical application include things like classification or figuring out diagnoses, regression or finding associations between things, predictions such as for the stock market or crop yields and other things, and generation or denoising, creating images which are either realistic or taking away artifacts from actual photos and cleaning them up. And similarly for audio, cleaning up or generating believable audio. But in healthcare, most of our work focuses on the classification problem or finding a diagnosis. And now there's also a lot of interest in predicting things, such as the effects of a particular treatment or a prognosis of a particular disease. Now, this is an interesting application of AI. Looking at this retinal photograph, no retina specialist, no ophthalmologist could figure out the gender of the patient. But the Google DeepMind project actually did that with very high reliability. And more importantly, they could look at this photograph and figure out the 5-year risk of a major cardiac event. So getting the terms right is kind of important because AI is thrown around in a lot of different areas. Artificial intelligence itself is just an umbrella term which covers all sorts of different subfields. Machine learning is primarily a mathematical approach to statistical parsing of data and improving on intelligent guesses over time. Deep learning is synonymous with neural networks and advanced machine learning. That's really where the current iteration of artificial intelligence is. So there have been a few different approaches to automated image processing over several decades. The crudest and original approach was a simple automated detector in which groups of pixels are defined as something and then the machine just goes out and looks for those. A more advanced approach is machine learning, in which guesses are made whether one group of pixels matches a target group that you're looking for, and over time, as the output or accuracy of those guesses is established, then the guesses improve over time. This is primarily a statistical method. And most recently, the deep learning or neural network model has really taken off and shown a lot of accuracy and power in figuring out elements within images. And this really loosely models the brain, in which the input layer, which is composed of pixels within the original image, is fed forward through different layers of the system, which are kind of like how neurons in the brain feed forward and either activate or make next layer neurons fire and so on. So using these systems, the inputs eventually cascade to an output, and that output is refined over time based on the accuracy from previous guesses. So this is where supervised and unsupervised learning come into play. Both use neural networks, but in the supervised approach, it is defined for the deep learning system what a hemorrhage looks like, what exudates look like, what vascular changes might look like, and then using the rules that we've defined for diagnoses for ourselves, then the machine puts them together and comes to an appropriate diagnosis. On the other hand, the black box model is a little different, where the system is shown images, and it is also given the final diagnosis without being told the rules of how to find that diagnosis. It's not really taught what hemorrhages look like or what other elements might look like. But after being shown many images, the system comes up with its own way of tying the images from their raw input state to the final diagnosis. The thing that makes black box learning a little bit unnerving to people in medicine is that, number one, there are no defined features that the machine has been shown or taught to recognize. We also don't know what labels it is actually internally creating, or what elements it's thinking mean other things. Having said that, the Google Brain Project correctly diagnosed diabetic retinopathy at least as well as general ophthalmologists. The problem, though, is with this black box model, with supervised learning, even when those things are defined, things that don't throw off a human grader, such as a few pixel changes in these sets of images on the left and the right, can throw off the machine and can get a very inaccurate result. So this is one of the big challenges in AI with regard to image recognition. Having said that, there are a number of different areas in which AI is being used. Those include fundus photography, or just color photographs of the retina, to recognize things like diabetes, macular degeneration, retinal detachments, and glaucoma. The OCT, which is a type of retinal scan, has also been used to diagnose macular edema or macular degeneration, and more importantly, to predict the kind of treatment burden that these things may require. So some of the notable systems that have been developed have come from some of the major players in computing, and these include things like IBM's Watson Artificial Intelligence Group. They actually classified diabetic retinopathy with a fairly high level of accuracy. The IDX system was developed at the University of Iowa, and that is actually now FDA approved as of 2018 to detect diabetic retinopathy requiring referral to an ophthalmologist. And Microsoft also has a system that they're developing for detection of diabetic retinopathy as well. There are several academic institutions working on OCT and fundus-based recognition of things like macular degeneration and diabetic macular edema. Some of these things have actually been portable and able to be transferred from ophthalmology into other areas of radiology. The Google DeepMind and Moorfield Eye Hospital have worked on a system which has broad application to a number of diseases, and this is actually a pretty major step forward, because these systems are typically good at classifying or making diagnoses within a specific subset of diseases, but not for a wide range of diseases. Then going back to that Google Brain Project, the really impressive one from 2016, where they looked at the fundus photographs and could predict the gender, they looked at 280,000 or so images, and importantly, they were able to figure out not just the gender, but also the age with a pretty high level of accuracy, smoking status, control of diabetes, even the BMI, which is incredible. There are vascular changes based on the very, very fine pixel level of images that we can't see as humans, that these unsupervised learning approaches may be able to uncover. So some of the goals of AI and automated image recognition in the future include things like diagnosing diseases besides just diabetes and macular degeneration, embedding these into devices so that more automated things can happen in primary care doctors' offices, integration into research centers so that doctors don't have to overread a whole lot of images, and then predicting or individualizing treatment for patients, not just using population studies, and finally predicting how much a specific treatment might be beneficial for an individual patient. But we're not the only field seeing the use of AI for image recognition. This has been around in dermatology for quite a while for diagnosis of melanomas. And now in GI, there are colonoscopies in which image recognition and looking for polyps is being done using AI methods. So in summary, I believe that deep learning is really going to change the delivery of healthcare. There is a lot of room and potential for discovery using unsupervised learning systems, and supervised learning systems will allow us to automate a lot of that care. Those disciplines which are primarily image-based, such as dermatology, radiology, pathology, and to some degree ophthalmology, these will probably be the ones that are first and more profoundly affected than some other areas in medicine. And these approaches may also allow for more personalized or patient-specific treatments and prognostic information in the future. Thank you very much for listening.
All right, thank you, Dr. Ahmad. We've a lot of good information today, so we're going to open it up. We're right at 7 o'clock now. We try to really respect your time, but it looks like to get some of our speakers in and some questions, this is going to go over a bit. We apologize for that, but I think it's important enough. If you're able to hang on for probably another 15 minutes or so, we'll get all your questions in. So we'd like to open it up to our panel of doctors for any questions that you might have. Take it away.
And just a few reminders, you guys can access the question and answer features at the bottom of your Zoom control bar. You can also use the hand raising feature and we will unmute your lines that way. So it looks like Rosemary has a question. So let's unmute you.
Hi, can you hear me?
Yes, we can.
Great, okay. You know, I am a patient of Dr. Connor at NCW and I love him. I wanted to ask a little bit more about is it a regular finding of fluid-filled cysts in the RP cells that is common with the degeneration of RP? And if that is so, do these approaches that any of you take toward analyzing RP and solutions to it, do any of them particularly zero in on that fluid fill cyst type of syndrome?
So I guess if nobody else wants to tackle that one, I'll speak up. I mean, this is basically a clinical observation from that retina specialists are fairly familiar with, that there is a type of structural cystoid spaces that can form in the retina when people have retinitis pigmentosa. And interestingly, it's not angiographically active. So unlike other processes where we do an angiogram, a fluorescein angiogram, and we can see the blood vessels leaking or actively exuding, we don't really see exudation in the same way, but you do get these structural cysts in the retina. Interestingly, there are some carbonic anhydrase inhibitors. This class of medicine that can that can actually reduce that, but that's unfortunately the only clinical treatment that we have at this point. It works in some people and it doesn't work in others. But on the scientific side, maybe some of our scientists can speak to that a little better.
Well, thank you for that.
Other questions?
Good evening. I'm Basir Ahmed from the Department of Ophthalmology at the Medical College of Wisconsin. I'd like.
I don't think anybody wants to hear that again. Definitely not me. Thanks.
There we go. Any other questions out there? Don't be shy. Oh, okay. I'll tell you what, let me, while you guys are collecting, if there's any others that you have, let me just finish my portion and then we can go back to that if there are any others. So let me just go here really quick. And I just wanted to get this in before you guys head out for the night. As I said earlier, we partner with MCW for many events throughout the year, but our big one is our Vision Walk. If I know there's a bunch of you on there that are team captains from our past events, but if there's anybody out there that hasn't done this before, obviously this year was pretty much a wash, but we did a virtual walk in May. It's usually in May every year at Veterans Park, and we're looking for families to come out and join us. The Eye Institute has a huge team that comes out there. They've been out there up to upwards of 115, 125 team members, along with a big group of the doctors that are out there. And it's great to see the connection between our families, our professional partners, our researchers, and our staff. And it's a great opportunity. We have a lot of fun. It's an important cause for us because we raise a lot of money, but we want to make sure it's fun for everybody and for people to keep coming back. So we try to avoid having hail and rainstorms, but that just sometimes doesn't happen. We try to avoid COVID-19, but we are hoping that we'll be back at Veterans Park this year. We have free everything. We have bounce houses, giant slides, tattoo artists, as you can see in some of the photos for those of you that are looking at the screen, or I'll describe some of those as well for those of you that can't. We have the mascots from pretty much every area of professional and college athletic team. We have Star Wars characters. My own South Milwaukee Lions Club serves our food. We usually do barbecue, grill, and a whole lot of fun. We have live music. So we're hoping that you can do that. There's a date set. Go to visionwalk.org under Wisconsin Vision Walk, and there's all the information there. We're still trying to figure out whether it'll be live. We're hoping that it is this year, but we'd love to have you out there. So if there's anybody that could join us and help us, it's free, but we'd love to help you fundraise so you can help us to give grants to some of these great doctors that you've heard today. So let me go back now. That's my closing statement, but I wanted to see if there's anybody else that has any other questions. We do. Kelly?
Okay, it looks like we have a question from Heather, so we're gonna unmute your line, Heather.
So this question is geared towards Dr. Ahmad and possibly Iris as well. I'm just thinking about the types of AI that we're using now for clinical use in ophthalmology. What types of needs are there now, or like, do doctors dream about being able to have the access to, by using like this AI technology, to identify and like diagnose or manage certain diseases, if that question makes sense?
Iris, is it okay if I go first? Sorry. Okay, so I think there are two parts to that question. The first is that the two approaches. One is unsupervised learning, where you have black box systems. You just feed them hundreds and thousands of images, and then you see what they're able to find or pull out of those. So that's really good for novel discovery. And the hope is that at our institution, as we're setting up neural networks and we're starting to get into this, that we may be able to discover something perhaps from AO images, adaptive optics images, things that are a little bit unique to MCW. And then the second part of that is using supervised learning approaches to kind of move the practice of regular clinical medicine forward. So in terms of, I think, what do we wish? One thing is for novel discovery, and that will be using black box, unsupervised learning models. And the second is supervised learning to kind of advance our regular day-to-day care and actually applying it to real patients. I hope that sort of touches on this.
And that sort of goes along the lines of big data too. And ophthalmologists have a big registry as well. It's called an IRIS registry, where it has national data from many academic and private practices. And sometimes you could gather data through that too, trends and stuff, because we have our patient populations, but sometimes you could figure out bigger patterns by looking at studies through the IRIS registry. And that's also kind of a fiscally expensive way to do projects, but it's very informative.
I would say one of the big limitations in AI is that you need really large data sets. Just like Iris is mentioning, you need, so big data really plays into it. You really need hundreds and thousands of images, for example, to find those novel or hidden things that might be rare. And this is actually one of the challenges why it's harder to apply AI to rare diseases like retinitis pigmentosa or other retinal degenerations, because there just aren't that many people that have it. And although academic centers may have bigger collections of those kind of images than other places, it's really hard at any one center to have enough of those images. So part of the move forward is to try to get centers to share images and to form these big data collaboratives so that they can pool all of their, for example, RP patients together, and then you can start to find some of those patterns that are hidden.
Yeah, so Dr. Ahmad was just talking about images and things like that, but data collection is just one thing I want to throw out there. The foundation sponsors something called My Retina Tracker. That's for data collection, and just something for you guys that are out there available to, we also have free genetic testing that we provide. So if you guys are interested in getting testing for your vision, that data also comes back to us through My Retina Tracker. And that's an opportunity for doctors throughout the country and throughout the world to approach us and see if there are people that might fit into the various clinical trials and research studies that the doctors today are going through. It's a pool of information from all of our members throughout the world. So if that's information that you'd like more information about that, please contact me and I can send you info on that as well. So just another opportunity for our families. Kelly, do we have any others?
Oops, sorry. This one was on the Q&A. It's for Dr. Kassem. For IOL in children, would you have to intermittently perform this surgery throughout their life? And if so, would it be beneficial to monitor the inflammatory process, not just the first time, but the other times the implant is done? I wonder if the inflammatory process would be reduced or if the inflammatory markers would change the second or third time this process is done as the body.
That's amazing questions, and that sort of summarizes a lot of our questions coming out of the lab, actually. So I'll go question by question. So for the IOL in children, so for the cataract surgery in children and adults, that's something that is just done once. So once you take, I lost the question. I'll go back to the question. So once you take the cataract out, it doesn't come back. Sometimes you'll hear about a secondary cataract, and that's more actually of that fibrotic process. And there are some cells that replicate, but the cataract doesn't return once you take it out. In children, if you put in the IOL, or in adults, once you put in the IOL, you typically try not to take it out or do that surgery again unless there's a complication. Excuse me. And in children, if you just remove the lens and don't put in the IOL, that would be a second surgery later on. And one of the things we look at is if you do things later on, or we look at later complications, because some of these complications can happen 10 years down the road. And so we look at the changes in these proteins short term, but also now we're looking much longer term, because there could be long-term shifts that could cause these complications. So yeah, there are lots of changes that happen just with aging, but also with every procedure you do, we believe that there are changes that happen and there could be alterations throughout life. I hope that answered your question. But that summarizes like my lifelong research questions, so.
Any others?
It looks like we have one hand up. Rosemary's hand is up. I don't know if that's from the last question or if you'd like to ask another one.
I think she has another one.
Actually,
Rose?
yeah, I do have a. Well, I was just amazed at the zebrafish and how tiny those eyes are and how much information you can get from that. And
Mind-boggling.
it is mind-boggling, beyond. And I guess I just wanted to know. But this is probably answered on that tracker. It's like, as a result of all these studies and findings, are there any more relevant? Testing that we can participate in?
From a zebrafish perspective, I would love to say yes. I'm working as hard as I can to make genetic models for as many human diseases as I can track down. I really agree with what Dr. Lipinski said, where he said that the goal would be to develop therapies that regardless of the genetic cause of the vision loss, you can offer a treatment. And so I would say things like that offer a huge amount of hope. Zebrafish are fantastic because they're so small and they develop very quickly, and we can use them to screen through libraries of compounds, novel drugs, things that may not yet be available for drug trials. And it allows us to whittle down, promising drugs that might be mobilized into animal trials and then ultimately human trials. So I would just say stay in touch with all of the great people who are coordinating fundraising here at the Eye Institute. Gina Fertachnik, who I neglected to thank at the end of my talk, is really wonderful. And if you were to reach out to her or Julianne Kelly, or really anyone at the Eye Institute, they could really direct you towards channeling all of your positive intents and fundraising our way. So that would be super if you were able to do that. That would be the most helpful thing of all, I would say.
Thank you so much for all the work you do.
Oh,
Thanks, Rose.
Yeah.
Any others? We'll take one more question if there's one out there.
Doesn't look like we have any other ones.
Okay. So I wanted to just sum up let me see if I can control my screen better. So once again, thank you to our 4 doctors who presented today, also for Dr. Kostakis as well, for joining us on this call. I know your time is precious and your weeknights, but it's important that our families get the information they can, and we love having you guys on board, and we appreciate the time. If you have any questions moving forward from any of us, whether it's the Foundation Fighting Blindness, as most of you know that know me, I'm available really 24 hours, 24/7, and you'll never be surprised. You should never be surprised if I answer you at 2 or 3 or 4 in the morning. Also, the doctors, and also as Julianne and Gina from MCW are available as well. Kelly from our office too. So if you have questions on anything that's going on today, if you want us to connect you with any of the doctors, if you can't get there directly, please contact us. Thank you all for joining us this evening, and you have a good night and stay safe and healthy. Thanks so much.
Thank you.
Thank you.
Thank you, everyone.
Bye-bye.
Have a good night.